First, I would like to thank Drug Delivery for inviting me to discuss Peter Chu formula for calculating COVID-19 vaccine efficiency. In this discussion, I emphasize the significance of educating the public about the importance of vaccination. By doing so, we can tackle the skepticism surrounding vaccines and improve vaccine acceptance. Article COVID-19 Vaccination Educational App highlights a major issue surrounding vaccine acceptance. People question the safety and efficacy of COVID-19 vaccines. Unfortunately, this skepticism has resulted in low vaccination rates, particularly among certain populations. It is crucial to acknowledge and address these concerns to ensure widespread acceptance and uptake of COVID-19 vaccines. By doing so, we can collectively work towards achieving higher vaccination rates and effectively combat the spread of the virus. Throughout this session, I will discuss strategies to promote better understanding and trust in the efficacy of COVID-19 vaccines. Together, we can pave the way for increased vaccine acceptance. Let's take a closer look at the findings of a survey conducted in Penang, Malaysia, which shed light on the reasons behind the refusal of the COVID-19 vaccine. The survey revealed that a significant number of individuals expressed concerns about the safety of the vaccine, accounting for 75.5% of the respondents. Additionally, 47.4% of the participants cited concerns about the effectiveness of the vaccine as a reason for their refusal. These findings highlight the need for effective communication and education to address these specific concerns. By providing accurate information and addressing misconceptions about the safety and effectiveness of COVID-19 vaccines, we can help alleviate these apprehensions and encourage higher acceptance rates. According to article World Health Organization, Field Evaluation of Vaccine Efficacy, Walter A. Orenstein et al. The ideal vaccine efficacy study is a clinical trial starting with persons susceptible to disease. In a double-blind randomized placebo-controlled fashion, half of the children receive vaccine and half receive placebo. To calculate vaccine efficacy, both groups are followed prospectively to determine attack rates for disease in vaccinees and non-vaccinees. This type of study is generally not possible after a vaccine has been licensed because the vaccine is of proven benefit, and use of a placebo is unethical. In most countries today, measles vaccine has been used in a proportion of the population. These vaccinees are a self-selected rather than a randomly selected group, and their susceptibility prior to vaccination is generally unknown. Nonetheless, vaccine efficacy studies are still possible if biases are reduced to a minimum in order to recreate as closely as possible the ideal conditions of the prospective clinical trial. General principles vaccine efficacy is measured by calculating the attack rates of disease among vaccinated and unvaccinated persons, and determining the % reduction in the incidence rate of disease among vaccinated persons relative to unvaccinated persons. The basic formula is written as vaccine efficacy equals attack rate in unvaccinated group minus attack rate in vaccinated group, and then all divide attack rate in unvaccinated group and times 100%. Let's calculate the measles vaccine efficacy in the Gambia for children aged 9 to 47 months using the given information. This calculation demonstrates that the measles vaccine in the Gambia has an efficacy of 89% for children aged 9 to 47 months. By comparing the attack rates between vaccinated and unvaccinated groups, we can determine the effectiveness of the vaccine in reducing the incidence of measles. The primary goal of the formula developed by Peter Chu is to simplify the calculation of vaccine efficiency. By breaking down the calculation process into easily understandable steps, individuals can grasp the concept more easily and engage with the information more effectively. It is crucial that we empower people to make their own assessments of vaccine efficacy. By providing them with a user-friendly tool, we encourage them to become active participants in their health decisions. This formula serves as a bridge, connecting scientific data with public understanding and facilitating informed choices. Ultimately, Peter Chu formula aim is to enhance vaccine acceptance. By demystifying the calculation process and offering a clear method for calculate vaccine efficiency, we can instill confidence in the public and promote higher vaccination rates. Let us empower individuals to make informed decisions and embrace the power of vaccination. Furthermore, the Peter Chu formula can assume a high target population of vaccination, such as the medical worker group, represented by a value of K equals 100. This assumption simplifies the calculation process even further, making it easier for individuals to grasp and apply. By simplifying a complex formula, The Peter Chu formula empowers individuals to engage in data-driven decision-making. It enables them to evaluate the efficiency of the COVID-19 vaccine and comprehend the impact it can have on their lives and communities. What sets the Peter Chu formula apart is its reliance on easily accessible data found in the news. It allows individuals to compare the average infection rates of both vaccinated and unvaccinated groups, enabling them to make informed assessments. This approach ensures that the formula remains transparent and accessible to all. The formula's simplicity aligns perfectly with Albert Einstein's famous quote, "Everything should be made as simple as possible." In addition, Albert Einstein's also quote, "1. We cannot solve our problems with the same thinking we used when we created them. 2. If you can't explain it simply, you don't understand it well enough. 3. Genius is making complex ideas simple, not making simple ideas complex. 4. Any intelligent fool can make things bigger and more complex. It takes a touch of genius and a lot of courage to move in the opposite direction. 5. God always takes the simplest way. 6. When the solution is simple, God is answering." Isaac Newton quote, "Nature is pleased with simplicity, and nature is no dummy." From the Albert Einstein's and Isaac Newton quote, it can be seen that simplifying knowledge is very important. For those interested in new simple knowledge, I recommend referring to my book titled Peter Chu Formula for Maximum Positive Rate Based on COVID-19 Mutant, 3rd edition. Simplifying Mechanical Engineering Solutions with Peter Chu Rule, Method and Theorem. Simplifying Electrical Engineering Solutions with Peter Chu Rule, Method and Theorem, and Simplifying Civil Engineering Solutions with Peter Chu Rule, Method and Theorem. The Peter Chu formula used to calculate the efficiency of the COVID-19 vaccine is similar to the sum of n terms and the sum of infinite terms geometric progression. In a geometric series, when the absolute value of the common ratio, r, is less than 1, raising it to the power of infinity will approach 0. Consequently, the sum of an infinite geometric series will converge to a value equal to the first term, a divided by 1 minus r. Similarly, in the Peter Chu formula, if we consider value k approaching 100, the vaccine efficiency can be calculated as b minus a, and then all divided by b and times 100%. Here, b represents unvaccinated infect rate, a represents vaccinated infect rate, and k% represents the percentage of a vaccinated group as compared with an unvaccinated group. By comparing these two formulas, we can observe the mathematical parallels between the convergence of a geometric series and the calculation of vaccine efficiency using the Peter Chu formula. According to article from Proofs in Mathematical Reasoning, Agata Stefanowicz et al., 2014. Mathematical proof is absolute, which means that once a theorem is proved, it is proved forever. Until proven, though, the statement is never accepted as a true one. Therefore, a mathematical proof of Peter Chu's formula is provided to ensure its soundness. Now, let's take a look at an example from a study published in the New England Journal of Medicine, titled Safety and Efficacy of the BNT162b2 mRNA COVID-19 Vaccine by Fernando P. Polak et al. 2020. This study provides valuable insights into the safety and effectiveness of the vaccine. During the study, a total of 43,448 individuals received injections, with 21,720 receiving the BNT162b2 vaccine and 21,728 receiving a placebo. Among those who received the BNT162b2 vaccine, there were 8 cases of COVID-19, compared to 162 cases among those assigned to the placebo group. First, let's put the data to a tablet. This table provides a concise overview of the distribution of individuals and the respective number of COVID-19 cases in each group, highlighting the significant difference in the occurrence of COVID-19 between the vaccinated and placebo groups. To calculate the vaccine efficacy based on the provided data, we can use the original formula. Vaccine efficacy equals attack rate in unvaccinated group minus attack rate in vaccinated group, and then all divide attack rate in unvaccinated group and times 100%. Given that the attack rate in the placebo group is 162 cases out of 21,728 individuals, the attack rate can be calculated as attack rate in placebo group equals 162 divide 21,728 times 100% approximately equals 0.7459%. Similarly, the attack rate in the vaccinated group is 8 cases out of 21,720 individuals, leading to attack rate in vaccinated group equals 8 divide 21,720 times 100% approximately equals 0.0368%. Using these values, we can calculate the vaccine efficacy. Vaccine efficacy equals 0.7459% minus 0.0368%. And then all divide 0.7459% approximately equals 95.0599%. Therefore, based on the normal calculation using the given data, the vaccine efficacy is determined to be approximately 95.0599%. This indicates a significant reduction in the incidence of COVID-19 among the vaccinated group compared to the placebo group. Indeed, it appears that when we apply Peter-Chew's formula, we obtain the same result for vaccine efficacy as the normal calculation. Both methods yield a vaccine efficacy of approximately 95.0599%. This consistency reinforces the validity and reliability of Peter-Chew's formula in assessing vaccine efficacy. The formula provides a mathematical approach to determine the effectiveness of a vaccine based on the number of cases in the vaccinated and placebo groups. In this particular case, whether we use the normal calculation or Peter-Chew's formula, we arrive at the same conclusion that the BNT162b2 mRNA COVID-19 vaccine has a high efficacy rate of around 95.0599%. This signifies a substantial reduction in the incidence of COVID-19 among those who received the vaccine compared to the placebo group. It's worth noting that the agreement between the two calculations further supports the soundness and applicability of Peter-Chew's formula in evaluating vaccine efficacy. When we use Peter-Chew's formula with a value of K equals 100, the calculated vaccine efficacy is approximately 95.0617%. This value is indeed very close to the normal calculation result of 95.0599%. The negligible difference between the two calculations further reinforces the reliability and consistency of both methods in determining the vaccine efficacy. Both approaches lead to similar conclusions, indicating a high level of effectiveness of the BNT162b2 mRNA COVID-19 vaccine in reducing the incidence of COVID-19. It highlights the robustness of Peter-Chew's formula and provides additional evidence for the reliability of both calculation methods. It is evident that all three calculations, normal calculation, Peter-Chew's formula, and Peter-Chew's formula with K approximately equal 100, yield nearly identical results for the vaccine efficacy. The fact that these calculations produce consistent results further strengthens the validity and reliability of the methods used to determine vaccine efficacy. It provides confidence in the accuracy of the assessments and reinforces the conclusion that the BNT162b2 mRNA COVID-19 vaccine has a high efficacy rate of approximately 95%. These aligned results from multiple calculation methods enhance the overall understanding and trustworthiness of the vaccine's effectiveness in preventing COVID-19. It underscores the importance of utilizing different approaches and methodologies to cross-validate findings, ensuring robustness and accuracy in scientific analysis. On November 18, 2020, Pfizer and BioNTech announced the completion of the Phase 3 study for their COVID-19 vaccine candidate, BNT162b2. The study results showed that the vaccine met all primary efficacy endpoints. The primary efficacy analysis revealed that BNT162b2 was 95% effective against COVID-19, starting 28 days after the first dose. In the study, a total of 170 confirmed cases of COVID-19 were evaluated. Out of these cases, 162 were observed in the placebo group, while only 8 cases were reported in the group that received the vaccine. These results are significant and demonstrate the vaccine's effectiveness in providing protection against COVID-19. The data indicates that receiving the BNT162b2 vaccine significantly reduces the risk of contracting the virus. Please note that this information is accurate as of November 18, 2020. If we consider a modification in the data where the vaccination process only reaches 90% of the total target group, we can adjust the vaccinated data accordingly. The original vaccinated data is 21,720 individuals. If we take 90% of this value, we would have 0.9 times 21,720 approximately equals 19,548. The original infect COVID-19 is 8 individuals. If we take 90% of this value, we would have 0.9 times 8 approximately equals 7.2. Even with the modification where the vaccination process reaches 90% of the total target group, the results from the three calculations remain almost the same. This further demonstrates the reliability, consistency, and robustness of Peter Chu's formula in assessing vaccine efficacy. Regardless of the vaccination coverage, whether it is 100% or 90%, the vaccine efficacy calculated using Peter Chu's formula remains highly consistent. This consistency highlights the inherent strength of the formula in evaluating the effectiveness of a vaccine in reducing the incidence of a specific outcome, such as COVID-19 cases. The fact that the results from the different calculations align closely affirms the validity of Peter Chu's formula and its practicality in assessing vaccine efficacy. It provides reassurance to researchers, healthcare professionals, and the public, reinforcing confidence in the formula's application and the conclusions drawn from its results. It is noteworthy that Peter Chu's formula provides a straightforward and accessible method to evaluate vaccine efficacy, allowing for reliable comparisons and informed decision-making in public health interventions. To learn more about Peter Chu's formula for calculating COVID-19 vaccine efficiency, you can find articles on reputable platforms like the World Health Organization (WHO) or Europe PMC. Europe PMC provides access to the full paper of the article. For a comprehensive understanding of the topic, I recommend referring to my book titled Peter Chu Formula for Calculate COVID-19 Vaccine Efficiency, 3rd edition. This book will provide you with detailed information on the formula and its worksheet. In a study published by the Journal of Infectious Diseases titled Vaccine Epidemiology, Efficacy, Effectiveness, and the Translational Research Roadmap, Jeffrey A. Weinberg et al., 2010, an alternative and equivalent formulation of vaccine efficacy is presented. According to this formulation, vaccine efficacy VE can be calculated using the equation VE equals 1 minus RR times 100%. Here, RR represents the relative risk of developing the disease for vaccinated individuals compared to unvaccinated individuals. This alternative formulation provides a different perspective on calculating vaccine efficacy, emphasizing the comparison of relative risks between the vaccinated and unvaccinated groups. It serves as an additional approach to quantifying the effectiveness of vaccines in preventing the occurrence of diseases. The relative risk, RR, is the ratio of the probability of an outcome in an exposed group to the probability of an outcome in an unexposed group. For example, in a study examining the effect of the drug apixaban on the occurrence of thromboembolism, 8.8% of placebo-treated patients experienced the disease, whereas only 1.7% of patients treated with the drug experienced the disease. Therefore, the risk ratio is calculated as 1.7 divided 8.8, which is 0.19. This can be interpreted as those receiving apixaban had 19% the risk of recurrent thromboembolism than did patients receiving the placebo. In this case, apixaban is considered to be a protective factor rather than a risk factor because it is associated with causing a reduced risk of disease. Assuming the causal effect between the exposure and the outcome, values of RR can be interpreted as follows. RR equals 1 means that exposure does not affect the outcome. RR less than 1 means that the risk of the outcome is decreased by the exposure, which can be called a protective factor. RR greater than 1 means that the risk of the outcome is increased by the exposure. Now, let's shift our focus to how the public can calculate the efficacy of COVID-19 vaccines using information from local news sources. For instance, there was a calculation conducted for Pfizer-BioNTech's COVID-19 vaccine efficacy, specifically for Malaysian healthcare workers. The news article, titled COVID-19 Outbreak in Malaysia, and published on January 25, 2020, reported the first case of COVID-19 detected in Malaysia. According to a news article dated February 5, 2021, it was reported that nearly 5,000 healthcare workers had been infected with COVID-19 so far, with 2/3 of them being women. Before the vaccination campaign began, a total of 4,756 healthcare workers had been infected. Now, let's calculate the average number of infections per day before the vaccination period. Assuming the start date as January 25, 2020, the day when the first case of COVID-19 was detected in Malaysia, and considering the time frame until February 5, 2021, which is approximately 376 days, we can determine that the average number of healthcare workers infected per day before vaccination was 12.65. Moving on to the vaccination progress, according to news released on March 17, the second dose of COVID-19 vaccinations was scheduled to begin. Based on the Centers for Disease Control and Prevention, CDC guidelines, full vaccination is considered achieved on April 1, 2 weeks after the second dose. A subsequent news report on April 17 mentioned that 9 healthcare workers were infected after being fully vaccinated. Considering the time from April 1 to April 16, which is 16 days, we can estimate that an average of 0.5625 healthcare workers were infected per day after being fully vaccinated. Note, the revised content has been further simplified and adjusted for clarity. It's worth noting that the calculation results align with a real-world study conducted on Pfizer-BioNTech's COVID-19 vaccine, which showed an effectiveness of approximately 95%. A ratio of 1 divided by the relative risk, RR, of 22.47 indicates that the risk of infection among the unvaccinated population is 22 times higher than that of the vaccinated population. According to a news report on February 5, 2021, a real-world study conducted among healthcare workers in Malaysia demonstrated that the Pfizer-BioNTech vaccine was 95% effective in preventing COVID-19. This finding aligns with the calculation results obtained using the Peter-Chu formula, further validating its accuracy. Peter-Chu formula calculation result also is consistent with the real-world study conducted by Pfizer-BioNTech's COVID-19 vaccine, and its effectiveness is about 95%. Referring to the table shown, if healthcare workers had not been vaccinated, it is estimated that approximately 202 infections would have occurred between April 1 and April 16. This means that 202 healthcare workers would have been infected during that period. Additionally, considering the previous data, an additional 193 healthcare workers would have been infected prior to April 1. These numbers are significant because infected medical staff can transmit the virus to vulnerable patients in hospitals. This transmission can result in potential complications and, in severe cases, even deaths among the patients. The importance of vaccination becomes evident when we consider the potential impact on both healthcare workers and the patients they care for. By reducing the number of infections through vaccination, we can protect not only the healthcare workers themselves, but also the wider community, including vulnerable individuals who depend on their care. These insights highlight the significance of widespread vaccination efforts and the role they play in mitigating the spread of the virus and preventing further harm. Considering that some members of the public may have limited computing power or lack the time to perform complex calculations, it would be beneficial to integrate the new Peter-Chu formula for calculating COVID-19 vaccine efficiency into user-friendly tools like the PCET Medical Calculator. The PCET Medical Calculator can serve as a convenient and accessible platform for individuals to input relevant data and obtain the vaccine efficiency results without having to manually perform the calculations themselves. By incorporating the Peter-Chu formula into the calculator, users can simply input the necessary parameters, such as the number of infections in vaccinated and unvaccinated groups, and obtain the vaccine efficiency output instantly. This approach reduces the burden on individuals to perform complex calculations manually, ensuring that vaccine efficiency can be easily understood and accessible to a broader audience. By leveraging technology and user-friendly tools like the PCET Medical Calculator, we can empower individuals to make informed decisions and better understand the effectiveness of COVID-19 vaccines without requiring advanced mathematical skills or extensive computational resources. On the COVID-19 related formula page, there will be 2 options available for calculating the vaccine efficiency. By selecting conventional formula option, users can calculate the COVID-19 vaccine efficiency using the conventional formula commonly used in vaccine evaluation. This formula may involve parameters such as the number of infections in the vaccinated and unvaccinated groups, and it provides a standard approach to assessing vaccine effectiveness. By selecting Peter-Chu formula option, users can calculate the COVID-19 vaccine efficiency using the Peter-Chu formula, which offers an alternative perspective on evaluating vaccine effectiveness. This formula considers parameters like the number of infections and provides insights based on the reduction in infections achieved by the vaccine. To learn more about vaccine efficiency and relative risk, users can click on the title, which will provide detailed information and explanations regarding these concepts. If users wish to exit the page or end the calculation process, they can click on the Buy logo, which will conclude the session or game. By offering these options and providing information about vaccine efficiency and relative risk, the COVID-19 related formula page aims to facilitate understanding and enable users to choose the calculation method that best suits their needs. On the Peter Chu formula page for calculating the efficiency of the COVID-19 vaccine, there will be two options available. Option 1. K not equal to 100 users can choose this option to calculate the vaccine efficiency using the Peter Chu formula when K is not equal to 100. Users can input the relevant data, such as the value of K, the number of infections in the vaccinated and unvaccinated groups, and obtain the corresponding vaccine efficiency result using the formula. Option 2. K equal to 100. When users select this option, the calculation becomes simple, because without the need to calculate the value of K. To return to the home page or main menu of the PCET calculator, users can click on the PCET calculator logo button. This allows users to navigate back to the starting point or explore other functionalities offered by the calculator. On the normal calculation page, the following options and features are available. Calculate button. After inputting all the necessary data for the calculation, users need to press the Calculate button twice to initiate the calculation process. Note: users should press the space where they want to input the number and then enter the corresponding value. By clicking on the title, users can access an example or sample calculation. This example provides a demonstration of how to input data and perform the calculation correctly, offering guidance and clarity for users. On the Peter Chu formula page for calculating the efficiency of the COVID-19 vaccine when K is not equal to 100, users will need to follow the instructions provided to ensure accurate input and calculation. Here are the steps. Key in the corresponding value for COVID-19 positive cases in the vaccinated and unvaccinated groups, and the value of K. Press Calculate 2 times. After entering all the necessary data, users should press the Calculate button twice to initiate the calculation process. These instructions and options are designed to ensure that users input the relevant COVID-19 positive case data accurately, perform the calculation by double pressing the Calculate button, access examples for guidance, and easily return to the home page for further use of the calculator. On the Peter Chu formula page for calculating the efficiency of the COVID-19 vaccine when K is equal to 100, the calculation becomes simple, as there is no need to calculate the value of K. Users should enter the corresponding value for COVID-19 positive cases in the vaccinated and unvaccinated groups. There is no need to enter the value of K, since it is already assumed to be 100. Press Calculate 2 times. After entering all the necessary data, users should press the Calculate button twice to initiate the calculation process. Workshop Peter Chu Formula for Calculate Vaccine Efficiency by Southeast Asian Minister of Education Organization. Regional Central for Education in Science and Mathematics, or CMOA-RECSAM. Theme: Education 4.0 Issues, Challenges and Future Direction towards CMOA Priorities and Sustainable Development Goal SDGs, Phase 2. Date: the 18th of May 2022. Venue: ICT Lab 1 CMOA-RECSAM, Penang, Malaysia. Collaborating partner: CMOA Secretariat, Bangkok, Thailand. The workshop explored convention formula and Peter Chu's formula to calculate vaccine efficacy. Peter Chu's formula provides a simplified method that allows participants to easily calculate and understand vaccine effectiveness. Comment from a participant of workshop Peter Chu formula, Dr. Chin Chi Keong.
Conventional formulas for calculating the efficacy of COVID-19 vaccines can be very complex. However, Peter Chu formula is so much easier. I agree that by using public news information, the public can easily calculate the efficacy of a COVID-19 vaccine using Peter Chu's formula. This is to ensure that the public can calculate and understand the efficacy of the vaccine for themselves, thereby convincing the public to get vaccinated against COVID-19. I agree that simple formulas like Peter Chu formulas for calculating the efficacy of COVID-19 vaccines are important to help the vaccine industry, and recommend organizing more Peter Chu formula workshops for vaccine industry experts to learn how to use Peter Chu's formula for calculating COVID-19 vaccine efficacy. I prefer to calculate the efficacy of the COVID-19 vaccine using the PCET medical calculator rather than the normal calculator.
By encouraging vaccination and implementing effective preventive measures, we can work towards reducing the burden on healthcare systems and saving lives. Let us continue to prioritize vaccination and take necessary precautions to ensure the safety and well-being of everyone.
Thanks for listening.